Chapter 3, Cellular Level of Organization. Cells are the smallest living unit in the body. The human body has about 75 trillion cells, about 200 different types of cells. The two general categories for types of cells are sex cells, which are your sperm and your ova. These are going to be haploid cells, which have half the amount of DNA that the other cells have. And then you have your somatic cells, which covers all other body cells. This includes all of the cells except the sex cells, and they are diploid and have twice the amount of DNA material. Your cell structure is going to be broken down into three main parts, and then we'll go into more detail on each section. The plasma membrane, which will separate the internal and external environment. The cytoplasm, which is all the intracellular material, and intra, I-N-T-R-A, means within, so within the cell. So within the cell, you're going to find the cytosol, which is the liquid portion of the cytoplasm. Then you will find organelles, which are intercellular structures, and there's a variety of different organelles, all working to keep the cell functioning. And then we will also have inclusions, which are large masses of undissolved material. Extracellular to the cell, which extra means outside the cell, will be the fluid. And this is going to be a watery medium that will surround the cell. And there will be differences between the extracellular and the intracellular fluids. The plasma membrane is very thin, about 6 to 10 nanometers in thickness, and it will be composed of lipids, proteins, and carbohydrates. The four general functions of the plasma membrane are: 1, to physically isolate the inside versus the outside of the cell. So it separates intracellular and extracellular fluids so they can be of a different consistency. 2, it will regulate the exchange with the environment. It's going to regulate what enters and what exits the cell, so we can maintain a difference between the internal and external environment. Sensitivity. This is the first part of the cell to be affected by temperature, pH, concentration changes, and will often respond to that. And the fourth is structural support. We have a variety of specialized connections between the cell membrane or between the cell membrane and the extracellular material that provide tissues with stability. The plasma membrane structure is made up of, again, the lipids, the proteins, and the carbohydrates. Here we're going to look at them in more detail. Your lipids, which are about 42% by weight of the membrane. The main lipid is the phospholipid bilayer. We talked about phospholipids when we were doing chemistry. These are going to be a diglyceride with a phosphate molecule. The phosphate head gives it a hydrophilic material, and the inside where the fatty acids are will make it hydrophobic. The heads will be oriented to the outside or the inside of the membrane, so think of it as sort of a sandwich where you've got bread on either side and then you've got fatty ham and cheese in the middle. So the bread is hydrophilic, where the ham and cheese would be, the inner layer would be hydrophobic. This is going to help to isolate the cytosol from the external fluids. Only certain things will be able to move across that membrane, and other things will have to have special ways to move them, so it's going to help to regulate the differences between the two sides of the fluid. The other type of lipid we have cholesterol plus some other lipids which will be embedded in the membrane. Cholesterol is necessary to keep the cell membrane what's called fluid, so it can change shape a little bit. You want to think of the membrane sort of as a thin layer of olive oil that is surrounding the cytosol. The membrane proteins, much more abundant, about 55% by weight, and there are two general classes. There are integral proteins. These are actually part of the membrane structure itself, and they will span the width of the membrane. And you can see in the photo above, in the picture above, that there are many of the integral proteins that are going all the way across the membrane. Then we'll have some that are peripheral proteins. These are on the outside or the inside of the membrane. Okay. They will have different jobs, and they're not nearly as abundant as the integral proteins. Now, proteins come in a variety of different types. There's a large number of functions. The first function is anchoring proteins, which will help to attach the cell membrane to the other structures, the organelles and other cell membranes, and stabilize its position. Okay. It will also attach to the cytoskeleton inside. Extracellularly, there will be protein fibers and other cells that it will also attach to. Recognition proteins, these proteins act as cellular name tags so that all of your cells have your first name on it. With myself, it's Debbie. So every cell I have is named Deb, Deb, Deb. So I can recognize those, and they also recognize where they're supposed to be. It's a liver cell or it's a kidney cell. The third function are enzymes, which are biological catalysts, which we spent some time on with chemistry. These are going to speed up all of the chemical reactions. Number 4 are receptor proteins. These are going to bind with an appropriate molecule, and ligand is a generic name for a molecule. They're going to respond to specific extracellular molecules, and then they may trigger a change in the cell activity once they bind with those. Like they might open a gate and let something in. Carrier proteins are going to carry molecules back and forth across the cell membrane because certain molecules can't get through that fatty layer. So they have to have a special way to get in, and the carrier proteins will do this, as well as your channel proteins, which will be large proteins that have a channel right through the middle that will allow certain water-soluble molecules to move through. They're highly specific, and they usually only allow one particular ion to move through it. So there might be a channel for sodium, there might be a channel for another ion, or water. Okay. And then we have our membrane carbohydrates. These are a small percentage, only about 3% by weight. And the carbohydrates are going to be components of complex molecules. They're going to be compound molecules. They're going to be made up of proteoglycans, which is the sugar with a little bit of protein, glycoproteins, which is protein with a little bit of sugar, or glycolipids, which is going to be with a little bit of sugar. And these are all going to form a layer on the outside of the cell membrane and extend to the extracellular layer, and they form a sugary layer. It's a coating called the glycocalyx. The glycocalyx is going to function for lubrication and protection of the cell membrane. Some anchoring and some ability for movement of the cell will be conducted by the glycocalyx. It will also serve as a binding, such as the glycoproteins and glycolipids service receptors, and for recognition, so that they are recognized, the cells recognized by the immune system. Your plasma membrane is selectively permeable. That means it's going to allow certain molecules to pass freely, very small ions, and restrict the passage of larger substances. You want to think of this sort of like as a window with a screen on it. If you close the window, it would be impermeable. If there was no screen, it would be freely permeable, and anything that could fit through that window would make its way in. But if we put the screen on, dust particles can get through and air molecules, but the sticks and stones can't make their way through it. So this is what your plasma membrane is, a selectively permeable membrane. To move materials into and out of the cell, we have 2 ways of doing this. We have passive transport, and this is when you are going to have a movement of molecules with the concentration gradient. That means going from a high to a low concentration, and this will use no energy. No ATP will be used at all. Active transport is the movement of molecules against the concentration gradient, going from low to high, and this will use ATP. We do both in our cells. So a way to think of this type of transport, if you were waiting to get inside to go to a concert somewhere, everybody's outside, the doors are locked, you can't go in. Once they open the doors, you could actually pick your feet up and put your arms on 2 people's shoulders and be carried right in with the flow. There would be no energy used at all. You'd be going from the high concentration where everybody was inside the building where nobody is, the low concentration. If you're halfway through and you realize you forgot something, you have to turn around and go back out. Now you are going against the concentration gradient because you're moving from where it's lower inside to higher outside, and you're going to have to use a lot of energy to push your way through that crowd. Passive transport is also referred to as diffusion. This is that directional movement to eliminate a concentration gradient, going from high to low and concentration and trying to reach equilibrium. Factors that are going to affect this rate of diffusion are going to be distance. The shorter the distance, the more quickly the concentration gradient will be eliminated. Molecule size. Smaller molecules will diffuse quicker. They can move quicker through the medium and go from point A to point B. If it's a bigger molecule, it takes longer to squeeze its way through. Think of that in a crowded room. If there is an adult and a kid, the kid will make it over to the far corner before the adult does. Temperature will affect it. The higher the temperature, the faster the diffusion rate. Gradient size, the greater the gradient. The bigger the difference in the concentration, the faster the diffusion rate. And electrical forces. If it's a negative interior of the cell, positive molecules will diffuse faster into the cell than negative ones will. Diffusion across the plasma membrane can occur in a few different ways. We have simple diffusion, and this is when small ions or molecules that are capable of passing through that lipid membrane, the ones that tend to be lipid soluble, fatty acids, steroids, alcohol, those small lipid soluble molecules can move right across the plasma membrane. They don't need any special way to get in. Okay? This also will include certain dissolved gases, oxygen, carbon dioxide, and water molecules. We'll have a little special way to do simple diffusion. Then we have channel-mediated diffusion. This is where the small ions then cannot readily move or diffuse across that lipid membrane. So they have special little openings, channels, protein channels, that allow them to go across. These are going to be limited by the size of the molecule, and they tend to be specific, and they may be slower than simple diffusion. But again, keep in mind, this channel-mediated diffusion is sort of like is still going to be diffusion. It's still going from high to low. It just opens a hole where the molecules can get through. Osmosis is a special biological term. It is the diffusion of water across a selectively permeable membrane. You have to include that selectively permeable membrane in this definition. Because diffusion of water, if it's in a beaker with grape juice on one side and water on the other, that's just diffusion. But when we're talking about the water diffusing across that membrane, it becomes a biological term. We have osmotic pressure of a solution, and this is going to indicate the force at which pure water will move into a solution as a result of a solute concentration. What that means, if there's more dissolved materials within the fluid, inside the cell, more water will move in quicker. If it's less materials, the water will move in slower. Okay? And tonicity is the solute concentration of a solution. So we can measure how much material is in that solution. And this is going to be a relative term. It's going to be relative to the tonicity inside the cell. Okay? So when we look at number one, we have three different possible ways of these solutions. The first one we're going to talk about is an isotonic. Now, iso means equal. So this means there's an equal concentration of solutes inside and outside of the cell. So there's going to be an equal amount of water inside and outside of the cell. And therefore, the water can diffuse across the membrane, but there will be no net change. It'll diffuse in, it'll diffuse out at the same rate. Okay, another thing you need to know is that the solutes are too big to diffuse in and out of the membrane. So the only way we can try to reach equilibrium between the solution and inside the cell is that the water has to diffuse or undergo osmosis into and out of the cell. In isotonic, there is no net change of water inside the cell, so the cell stays the same. And in this figure we have, it's a red blood cell. It is naturally a biconcave cell, so it looks sort of like a little donut. This is the way your cells want to look, your red blood cells want to look. With a hypertonic solution, hyper means more than, so there are more solutes in the solution than inside the cell, which means that there's more water inside the cell than outside, because we want to have it equal 100% solution. The water, therefore, will move out of the cell, trying to dilute the solution that is surrounding the cell, and it's going to do this movement via osmosis. But the cell itself, when we're looking at red blood cells, will undergo some shriveling up, and because it's losing fluid, and this is called crenation, and this can have a huge damaging effect on your red blood cells. This is why if you become in a situation where you're dehydrated, the water will leave the cells, and this will cause death within 3 days. With a hypotonic solution, hypo means less than, so there are less solutes in the solution and more solutes in the cell, which means that there's more water outside the cell this time, and so the water is going to diffuse into the cell. It's going to diffuse via osmosis into the cell. It will cause the red blood cell to now swell and plump up. It's no longer that biconcave figure that we want it to look like and when it's in an isotonic solution. And it could, if it takes in too much water, could rupture. And when a red blood cell ruptures, it's called hemolysis. Now, if you are dehydrated, you go to the hospital, they're going to give you IV fluid that is isotonic to try to get your red blood cells back into that biconcave shape rather than the shriveled crenated shape. The next type of transport requires carrier proteins that are in the membrane to move the molecules into and out of the cell. These are called carrier-mediated transport, and they can be passive or active processes. Okay. The carrier protein will bind with a specific ion or organic substance and carry it across the membrane. Okay. Characteristics of your carrier proteins: they have specificity, which means that one carrier protein will only bind with a specific molecule, a certain molecule. So you'll have carrier proteins for, in this case, glucose, but you'll have carrier proteins that bind with other molecules also in the cell membrane. Saturation limits. This is that the availability of the carrier proteins and the substrate molecules will limit the rate of this carrier-mediated transport. Think of it this way: if there's only one doorway to get in and there are 10 people in line and only one person can go through the door at a time, it's going to go slowly. The only way we're going to speed that up is if we open more doors. So maybe we open 10 doors and everybody gets to go in at the same time. If, however, you need to have 2 or 3 people before we open the door and there's only one person standing in line, that's the substrate, you're going to wait a while until the other ones come before the door will finally open and let you in. So this is where the number of carrier proteins and the number of substrate molecules will regulate and limit the rate of the carrier transport. Regulation, the third characteristic, is the binding of other molecules, such as hormones, can affect the rate. They can bind certain molecules that will not allow the carrier protein to open up to accept the molecule it wants to allow through. We have, under carrier-mediated transport, both active and passive processes. So the first one we're going to talk about is facilitated diffusion. This is a passive process. It's diffusion, which means it's going to move molecules from a high to low concentration. However, it is facilitated because if you didn't have this carrier protein, those molecules couldn't get across the membrane. So we aren't going to be able to do anything different than moving it from high to low. We're not going to reverse the flow, but this facilitates or helps those molecules to get across that membrane by providing an opening to the other side. Okay. There's never a continuous open channel inside and outside the cell. It opens and closes, so it will regulate the rate at which molecules can move in. Continuing with carrier-mediated transport, we're now going to look at active transport. This is going to be against the concentration gradient. It's going to use ATP to move the molecules. We have ion pumps that actively transport cations. These are positively charged ions such as sodium, potassium, and calcium across the plasma membrane. And some of these ion pumps will actually be exchange pumps where they will move different molecules in different directions at the same time. The most common of these pumps is the sodium-potassium exchange pump, and this will move 3 sodiums out of the cell and 2 potassiums into the cell at each time it functions. And this carrier protein is also called the sodium-potassium ATPase pump. The thing is that sodium has got a lot more sodium in the extracellular fluid, and you have a lot more potassium in the intracellular fluid, and those will constantly diffuse out, but we constantly have to pump them back so that we have it sodium outside, potassium inside, so the cell can function properly. We need that non-equilibrium between those two molecules. Another type of active transport is going to be vesicular transport. Vesicular is, or vesicle, is a large empty sac. We'll fill it up with material and then we'll move it into the cell. These are going to be moving materials that are not just small molecules, but bigger molecules or bigger substances that can't just move across the plasma membrane. We're going to move material into the cell through endocytosis. Endo means into, and cyto refers to cell. So you're going to move material into the cell as seen in the diagram above, where it's starting with number 1, where it's going to start to pick up molecules, engulf them, and then put them in a little package, which is the vesicle. Okay. Receptor-mediated endocytosis, which is being shown in number 1, is when specific molecules bind with the receptor on the membrane, and this triggers the membrane to engulf the molecules. They're then going to be packaged up into vesicles and moved into the cell. This is going to allow for a concentration of specific molecules within the cell. If we need a lot of one type of molecule in there, we can do this by binding them to the receptors and engulfing them. Okay. Even though there may be a low concentration in the extracellular fluid, we're binding and gathering them all up in one little spot and taking them in. Usually glycoproteins are the receptors, and the ligands, the things they bind to, will be hormones, insulin, iron, transport proteins are all examples of what they will bind with. In addition to receptor-mediated endocytosis, we have 2 other ones based on the engulfment of particles into the cell. The difference will be whether this particle is small or large, whether it is dissolved in the fluid or is undissolved. And these are going to be pinocytosis. Pino in Greek means cell drinking or drinking. With the cell drinking, we're going to have the engulfment of a small dissolved particles, so the cells will be absorbing nutrients, say in the small intestine this way. Phagocytosis, and phago means to eat, is cell eating, and this is going to be the engulfment of large undissolved particles, such as a macrophage, which is a type of white blood cell, engulfing a bacteria to kill it. Okay. Both of these will be occurring continuously throughout the cell so that we can move material into and out of the cell. You can think of this if you think of pino versus phago. If you eat an orange, chewing it up, you're getting phagocytosis. If you're drinking orange juice, you're doing pinocytosis. You're taking in the nutrients, but they're in a liquid form. Once you've gotten material into the cell, say if you're a macrophage and you engulf the bacteria, break it down, now you have a waste product. You want to spit that out of the cell. That is going to also be put into a vesicle, and that will be spit out through exocytosis. The vesicle will come to the surface of the cell membrane, fuse with the membrane, and discharge all of the waste products out into the extracellular environment. We also will see this done with secretory products such as mucus and hormones when they are produced and released from the cells. Now we have finished looking at the plasma membrane, its structure and function, so we're going to turn to the remaining parts of the cell structure. We're going to talk about the cytoplasm, which contains the cytosol, which is the intracellular fluid, and then the organelles. With the cytosol, this intracellular fluid is going to contain dissolved nutrients, ions, some soluble and insoluble proteins, and waste products. There will be distinct differences between the cytosol, the intracellular fluid, and the extracellular fluid that surrounds the cell. The concentration of potassium will be higher in the intracellular fluid than the extracellular, and the concentration of sodium is lower in the intracellular fluid than is in the extracellular fluid. The intracellular fluid is also going to contain a high amount of suspended proteins, such as enzymes and other protein structures. The intracellular fluid usually contains a small quantity of carbohydrates and a large reserve of amino acids and lipids. And there may also contain inclusions within the cytosol, which are masses of insoluble material. This is the most common form of stored nutrients. The second part of the cytoplasm is the organelles. The organelles are intracellular structures that are going to be suspended within the cytoplasm and within the cytosol. And they're going to have a variety of different functions. There's going to be two divisions of organelles, non-membranous and membranous organelles. Your non-membranous organs are not completely enclosed by a membrane, so all of their components are in direct contact with the cytosol and whatever's happening in the cytosol. These are going to include your cytoskeleton, microvilli, centrioles, cilia, ribosomes, proteasomes, and flagella. Your membranous organelles are going to be isolated from the cytosol by a phospholipid membrane. Might vary a little bit from the phospholipid membrane of the cell plasma membrane. These are going to include your endoplasmic reticulum, both smooth and rough, Golgi apparatus, lysosomes, perioxysomes, mitochondria, and nucleus. Starting with the non-membranous organelles, we're going to start with the first group called the cytoskeleton. Now this is going to be a dynamic network of strands, filaments, fibers of protein structure that are going to form an internal skeleton of the cell. There are three different types of protein structures involved. We have microfilaments. These are very small, thin in diameter, only 6 nanometers. And a filament is a solid structure, sort of think of it as like fishing line. Okay. These are going to be made up of actin, which is the protein, and they function to anchor the cytoskeleton to the integral proteins of the plasma membrane, determine the consistency of the cytoplasm, and also are involved with the movement of the cell. The second type of cytoskeleton filament is the intermediate filaments. These are a little bit larger, 7 to 11 nanometers, and they function to strengthen and maintain the shape of the cell membrane as well as to stabilize the positions of the organelles in the cell with respect to surrounding cells. They're going to do this by attaching to other cell membranes or attaching the organelles to the cytoskeleton. And the third protein structure is a microtubule. This is considerably larger in diameter, 25 nanometers, but this is a hollow tube. You can envision this sort of like a straw. And it is going to function to provide strength and rigidity to the cytoskeleton, anchor organelles also to the cytoskeleton, cell movement and organelle movement within the cell. It will play a large part in cell division, mitosis, when we talk about that, and the movement of the DNA within the cell during division. The second of the non-membranous organelles are microvilli. Now these are small finger-like projections of the plasma membrane. They contain microfilaments going out to give them some support, and their function is to increase the surface area of the cell membrane for absorption, so they can absorb extracellular materials. They're going to be found lining the small intestine to absorb nutrients. Not every cell will have this particular organelle, only certain ones that have specialized absorption characteristics. This figure is just showing the internal structure of the microvilli, so you can see the microfilaments that are part of the cytoskeleton going out to give support to the microvilli. Here we have the third of the non-membranous organelles, the centrioles. These are two little organelles that are found in the centrosome, which is the zone where they live. Think of it as their neighborhood. And they're going to form the spindle apparatus, which is going to be used for cell reproduction when the cell divides the DNA. These are not found in mature red blood cells, muscle cells, skeletal or cardiac, or typical neurons, which means that they can't divide to make new cells. So once they die, you lose them. You can't replace them. The fourth type of non-membranous organelle are the cilia. You can see them in the top picture to the right hand. These are long, slender extensions of the cell membrane. Compare those to the larger picture to the left, which has just a stubby little microvilli. that are used for absorption. These long, thin cilia, which are composed of microtubules, are going to be used for two different things. Primarily, they act as sensors, and secondly, to the outside world, and secondly, they will work as motile cilia, where they can move material across the cell surface. You're going to find these in your trachea, where it's trying to move debris. If you walk into a dusty room and you swallow or breathe in the dusty air particles, they will be caught in the mucus that you produce, and then the cilia will work to push the mucus back up towards your mouth so you can cough it out. The fifth type of non-membranous organelle are the proteosomes. Your proteosomes are going to be these hollow cylinders that contain protein digesting enzymes. Their function is to break down and recycle any damaged or abnormal proteins that are found in the cytosol. Your cells are the best recyclers. Anything that is old and worn out, we will break it back down and recycle the materials back to produce new materials. Number 6 are your ribosomes. These are going to be made up of protein and RNA, the ribosomal RNA that we mentioned earlier. They are going to function in protein synthesis, and they can be either freely floating in the cytosol or attached to the rough endoplasmic reticulum. When they are attached to the rough endoplasmic reticulum, those are called fixed rhizomes. Now, they're going to make proteins no matter where they're found, but the proteins, the ribosomes that are fixed to the rough endoplasmic reticulum, are going to make proteins that are going to be exported out of the cell, or at least exported to the plasma membrane, where the ribosomes that are free in the cytosol are making proteins that stay within the cell. And number 7 is a flagella. This is a whip-like tail, we don't have a picture of it here, that is found only on sperm in the human body, and it's used for propulsion. We are now going to start looking at membranous organelles. These are going to be surrounded by phospholipid membranes that often are originating from the nuclear membrane, so they're an extension from the nuclear membrane outward. The first membranous organelle we're going to look at, which is also the 8th organelle of the cell in general, is the endoplasmic reticulum. This is a fancy word. Endo means inside, plasmic means in the plasm, and reticulum is a Latin word for webbing. It looks like spider webbing spreading out throughout the cytoplasm. This is a network of intercellular membranes that are connected to the nuclear membrane, and they form channels that are called cisternines as they move out into the cytosol. And we are going to have these associated with both ribosomes and not associated with ribosomes. The rough endoplasmic reticulum, RER, will have ribosomes attached to it. Recall the ribosomes make the proteins. They're going to make like a generic kind of protein, and then they're going to put it into the RER, and as it moves down through this channel, the cisternine, it will be exposed to a variety of enzymes that will modify this protein and change it. Okay? Then that will be packaged up and sent to the Golgi apparatus. Now think of it this way. If we all started with the same car, same basic car, and then we put it in this channel, and I wanted a pink car and you wanted it to be yellow with flames on it, and somebody else wants a sunroof, we can start with the same basic car but modify it so they all look a little different. When we're done, they can be used for a different function. The smooth endoplasmic reticulum, which will be after the rough endoplasmic reticulum, and some of the proteins will be sent there. Some will not. Some will be immediately just go through the rough and then be sent to the Golgi to be sent out of the cell or to the cell membrane. But some will be sent to the smooth. Keep in mind, your DNA only gives you directions to make proteins. It doesn't tell you how to make lipids or carbohydrates. So the way our cell works it is that we make the proteins, and then we're going to send them to the smooth endoplasmic reticulum, and we are going to then modify them and change those proteins completely into different types of carbohydrates or lipids. We are going to use our smooth endoplasmic reticulum, which has no ribosomes, to synthesize phospholipids and cholesterol for the plasma membrane. It's going to synthesize all our steroid hormones, such as testosterone and estrogen. It's going to synthesize triglycerides. Again, this is another type of lipid. It will form and store glycogen, which is the storage form of glucose. And it will form these in skeletal muscle cells and liver cells is where this will occur. It's going to store calcium in our skeletal muscles. This is a special job. And it will also detoxify drugs in the liver and kidney cells. Now, number 5 and 6 are very specialized. Different organs will have their smooth endoplasmic reticulum do different functions. The next membranous organelle is the Golgi apparatus. This is going to be made up of 5 to 6 flattened discs with channels, again, the cistern inside, and we're going to move material through it and produce 3 different types of vesicles. Vesicle, again, is going to be a sac that is going to then be filled with something and move it. We have secretory vesicles. These are going to be modify and package up secretions for release from the cell via exocytosis. So these will be filled with whatever the product is that will then go to the cell membrane and then will be secreted out of the cell when it ruptures. We have membrane renewal vesicles. These are going to be packaged with secretions that go to the membrane to renew and modify the membrane because the membrane is continuously changing. This continuous change is called membrane flow, where the vesicles bring over new materials, lipids, proteins, carbohydrates, and then they get released into the membrane. This provides a mechanism for cells to change characteristics of the plasma membrane as cells grow and mature and respond to the environment. And the third type of structure is a lysosome. This is going to actually be an organelle on its own, and this is going to be a vesicle that's filled with digestive enzymes that we will discuss on the next slide. The next membranous organelle is called the perioxisome. A perioxisome is a vesicle that's filled with digestive enzymes whose function is to break down fatty acids. These fatty acids are going to be broken down to hydrogen peroxide, and hydrogen peroxide is toxic. So the perioxisomes have a second set of enzymes to break down the hydrogen peroxide to water and oxygen to decrease the toxicity. Now we're looking at the lysosomes again. These are an organelle that is a vesicle that contains hydrolytic or digestive enzymes. These break down large substances. Old organelles, foreign invaders, this is what the macrophage would use to attack bacteria that it engulfed. And it will release all of its waste product via exocytosis to spit it back out of the cell. With your lysosomes, they will exist originally in the cell as a primary lysosome. This is filled with inactive enzymes because you don't want it to start to digest the cell itself. When it fuses with the substance to break down, it becomes a secondary lysosome, and the enzymes are activated, and then it can digest it, break it down, and then spit the waste product out through exocytosis. Now, we've talked about so far a number of different organelles that are involved with digestion. We have the lysosomes, which are doing large substances on old organelles. We have the perioxisomes, which are digesting fatty acids, and then have a second set of enzymes to break down the hydrogen peroxide, which is the metabolic waste product from digestion of fatty acids. We also had the proteosomes. The proteosomes are the ones that are digesting old proteins. So there's a lot of digestion and recycling occurring within the cell. The next membranous organelle is the mitochondria. These are going to produce ATP via cellular respiration, which we will spend chapter 25 on cellular respiration, how that is done. The mitochondria is a double membrane organelle. It has an inner and outer membrane. And this is believed to have been one of the first organelles that developed within a cell. It was engulfed, the primitive cell engulfed bacteria that then stayed within the cell and multiplied and continued to do its own job. And the mitochondria, the inner membrane has folds on it called cristia, and these function to increase the surface area for ATP production. In addition, your mitochondria has its own DNA, so it can increase the number of mitochondria by undergoing division and replication on its own. The largest organelle in the cell is the nucleus, and this is the control center for the cell. It's going to house the genetic information in the DNA, and the DNA is going to direct protein synthesis. The nucleus is going to have a lot of different structures associated with it. It has its own nuclear membrane called the nuclear envelope, and this nuclear envelope has holes in it called nuclear pores, so ions and small molecules can enter and exit the nucleus. It has its own fluid, the nucleoplasm, which will contain ions, enzymes, RNA and DNA nucleotides, plus some RNA and DNA. And it has its own matrix. This is in the nucleoplasm, similar to the cytoskeleton. It has a network of filaments for structural support, and it has its own organelle, the nucleolus. Nucleolus is singular, nucleoli is plural. Usually there are 2 to 4 of them within the nucleus, and these are transient nuclear organelles, and they make ribosomes that are used for protein synthesis. Within the nucleus, we find our DNA, and DNA has that double helix structure, and it wraps around proteins called histones. You'll have a group of 8 histones that the DNA will wrap around, and that group of 8 plus the DNA is called a nucleosome. Think of this, I know you guys probably don't know anything about old-fashioned phone cords, some of you might, but they were coiled cords, so you can think of this sort of as this coiling as a phone cord, and in between the phone cord, inside it, will be little balls of protein. When the DNA is relaxed and stretched out, it has a different name. It's called chromatin. And this is when the cells are not dividing. When the DNA condenses, it is now called chromosomes, and this is only going to be formed into the chromosome structure when the cell is dividing. It's the same material, it just has 2 different structures. Sort of like when you're working, you wear one outfit. When you have your days off, you have your more relaxed clothing on. Okay? Your chromosomes are made up of genes. So along a chromosome, you have 23 pairs of chromosomes. You will have a number of genes on each chromosome. These are going to be the functional units of heredity. They're going to dictate protein synthesis. And your genes are made up of the nitrogen bases, which is what your DNA is made up of, or nucleotides. And a group of 3 nucleotides or 3 nitrogen bases is called a codon. And we're going to read that code, sort of like a little genetic code. We're going to read a group of 3, and that will trigger an amino acid to come over and tell us what amino acid to add to the protein. Then we'll read the next 3 and the next 3, and each one will bring over an amino acid, and that's how we take the genetic information of DNA and convert it to the amino acid information of a protein. We're now going to look at how protein synthesis does occur. This is going to involve a number of players. It's going to involve your DNA, which provides the information for protein synthesis, the messenger RNA, which will copy the information for protein synthesis, ribosomal RNA, which will provide a location for protein synthesis, and the transfer RNA, which will bring over the amino acids when they're called for. The first part of protein synthesis is called transcription, and transcription means to make a copy. This is when the DNA information is copied onto the messenger RNA. This is going to be done by the DNA unwinding in a specific area, and when we're doing this, we're only going to copy like 1 gene, not the whole strand of the chromosome. We're only looking at a particular area in that chromosome where that gene is located that we need that protein to be produced. So the DNA will unwind and the 2 strands of DNA will separate. Then a strand of messenger RNA will form a complement to the DNA. That means it's going to copy the DNA, the reverse order. So where there was an A on DNA, a U for messenger RNA will be put on the messenger RNA. Where there was a C on DNA, it will be a G put in place on the messenger RNA. And where there is a T on the DNA, you will place an A on the messenger RNA. Remember that your messenger RNA does not have thymine. It does not produce that. It uses uracil instead in the place of thymine. That first strand of messenger RNA that's produced is called a pre-messenger RNA. And this first copy will contain both introns, which are non-coding regions, and exons, which are coding regions, which will actually code for the protein. So we have to do some splicing here. We're going to splice out all the introns from the pre-messenger RNA because they're just junk. We don't need that. And then we make a strand of only exons, all coding information that will code for protein synthesis. Now this is all being done within the nucleus where the DNA is housed. It can't leave the nucleus. But the messenger RNA is considerably smaller, and once it forms a nice strand of just exons, it will leave the nucleus through the nuclear pores and will go out into the cytosol. The second part of protein synthesis is translation. In this case, we're going to take that nucleotide information and translate it into a amino acid information so we can make a protein. We're going to utilize here the messenger RNA. Again, this has the code on it, the message from the DNA. This is going to come out, it's going to bind with a small subunit of the ribosome that's in the cytosol, and then there will be a second subunit that will form on top of it. The messenger RNA will be read and codon, so those three nitrogen bases. So the first one on this figure is AUG. That will be a start codon and say, okay, this is where we're going to start transcribing this information. Then we'll read the next codon. Now within the ribosomes, we have two little seats. Think of it for two codons to fit. So it'll move in, and that first codon will be read, and that codon will call for a transfer RNA to come over. The transfer codon RNA has an anti-codon, the complement codon. So in the first one we see AUG, the transfer RNA will have UAC, and that will come over, it will match up, and that transfer RNA not only has the anti-codon, so it comes over at the right time, but it also is bringing over a certain amino acid, and every single transfer RNA with UAC will bring over the same amino acid. Then we'll have the next codon in the next seat read, and another transfer RNA will move in, and those two will then be covered with the top of the large ribosome unit, and they will be trapped there. Now looking at the figure, we have the small ribosome subunit, the messenger RNA, the two seats in the large ribosome unit, and there will be two transfer RNAs, one right next to the other. When they both sit in those two sites, now we're ready for the codon, the transfer RNA in the first, to relocate and move. So in the next figure we'll look at that. So again, we're going through this in short steps. Here we have the two transfer RNAs sitting in the two hot spots in the ribosome. They each have the transfer RNAs have the anti-codon that complements the codon of the messenger RNA. They both had originally an amino acid they brought over, but now the first transfer RNA that was to arrive, that's in the middle unit, is ready to leave. So it doesn't want to take its amino acid back with it. So it hands it to the amino acid on the second transfer RNA. They form a peptide bond, and we are now starting to build the protein. The transfer RNA that was in the first, that arrived first in the middle section here, will then leave. Here in the diagram, you can see the first transfer RNA exiting the ribosome, and then the ribosome jumps over, and now there is a new codon in the second space, labeled, the code space is labeled A. Here comes another transfer RNA, and we're going to settle in, so you'll have two transfer RNAs, one in each space. The first transfer RNA is holding amino acids 1 and 2, the second one has amino acid 3. Now this process is going to continue. So the transfer RNA that had amino acid 1 and 2 hands it to transfer RNA with amino acid 3. It will leave, the ribosome will jump over, a new transfer RNA will come in, and then the transfer RNA with 1, 2, 3 will hand it to number 4 amino acid. And this will continue until you hit a stop codon, at which point the ribosomes release the messenger RNA, and the chain of amino acids, which is now a polypeptide or possibly even a protein, will be released. The next few slides are going to be a review of the protein synthesis, just a little quicker. The DNA will copy the information to the messenger RNA. This occurs in the nucleus, and those are the only two people involved in that process, and that is called transcription. The messenger RNA will then go into the cytosol, hook up with the ribosomes, and start making the proteins. This is going to involve ribosomal RNA for providing the location, which is the ribosomes, and transfer RNA, which will bring over the amino acids. And this process is called translation. Whereas transcription took place in the nucleus, translation will occur in the cytosol, and that messenger RNA can attach to either free ribosomes, where they're going to make proteins that stay within the cytosol, or they can attach to a fixed ribosome, and in that case the protein will then enter into the rough endoplasmic reticulum, be modified, packaged up, sent to the Golgi or possibly the smooth to be further modified, and ultimately be secreted from the cell, or it will be part of the plasma membrane. Again, the proteins made by the free ribosomes will stay in the cytosol for that individual cell's use. And proteins made on the fixed ribosomes, which are attached to the rough endoplasmic reticulum, will enter into the rough endoplasmic reticulum, the RER, and they will fold into their proper shape, and then these proteins will exit the cell and become part of the plasma membrane or outside of the cell and be carried somewhere else as a secretion. The proteins will be modified in the RER, then they're going to be packaged and sent to the Golgi apparatus for those to then be packaged up to be sent out of the cell or to the plasma membrane. Some proteins will be made by the RER and then sent to the smooth endoplasmic reticulum, where they will be further modified into lipids and carbohydrates. Here we can see the packages of proteins arriving at the Golgi apparatus. They arrive on one side and then they will be moved through it and spit out on the other side. We are seeing the proteins now moving through the Golgi apparatus to the opposite side, where they will be packaged in secretory vesicles, possibly into lysosomes as digestive proteins, or into membrane flow proteins, where they will be carried to the membrane. This is just an up close showing the Golgi packaging some of those hydrolytic enzymes into the vesicles called lysosomes. And this is showing the other packages that the Golgi will produce, the secretory vesicles that will be released via exocytosis, and the membrane renewal vesicles, part of membrane flow, that will carry proteins to the plasma membrane and replace the older proteins of the membrane. Now we are going to look at the cell's life cycle. You want to think of it just like you have a life cycle. You're born, you grow, you reach a point where you can reproduce and mature, age, and die. Your cells will do the same thing. They're going to grow initially, then they'll mature and reproduce, and they will die. When they die, a natural death, it is called apoptosis, and this is a pre-programmed cell death, versus necrosis is cell death due to injury or disease. Now, your cell life cycle is going to be broken up into a number of different phases. It's going to be broken up into two big phases, interphase and then mitosis. Interphase is when you're going to be doing normal growth, metabolism, and preparation for cell division. Think of it as inter means between. It's between the actual division phase. Okay? And during this phase, interphase, it will be broken down to smaller subphases. We have G1, which is going to be normal growth and metabolism. We're going to duplicate the organelles, and we're going to do protein synthesis. And this is where we spend the majority of interphase. The majority of the cell's life is in G1. Then it will enter into S. S stands for synthesis, and this is when the DNA is synthesized. We're doubling the DNA now so that we can reproduce, and we can give each of our offspring the same amount of DNA and the same exact genetic makeup that the parent cell has. So if you think of it as cookies, if I had one chocolate chip, one peanut butter, and one M&M cookie, now I double it so I have one of each of those, so I can give each of my daughter cells one chocolate chip, one peanut butter, and one M&M. So they are identical to what I was like to begin with, and they are each identical to each other. Right? DNA synthesis, that means we're doubling the DNA. Then we enter into G2. Now, once you've entered into S, you can't turn back. You have to divide. So G2 is just going to be the final prep for getting ready for this division. We're going to finish protein synthesis. We're going to complete the centriole replication because we need the centrioles to separate the DNA, and we need to have enough to give to each offspring of the centrioles. Some cells will enter into what's called GO, and that is they skip G1, S, and G2, and they live their whole entire life in GO. And this is a cell that is not ever going to divide. They're not preparing for division. They're just going to live normally, grow, metabolize continuously, and then die, but they don't reproduce. These will be your neurons, your skeletal muscle cells, your red blood cells once they hit maturity. Okay. After interphase, we're going to enter into mitosis. Mitosis is going to be cell division. And mitosis is asexual cell division, which means it's cloning. When we are going to do DNA replication in the S phase, we are going to have to unwind the DNA completely. So now you have 2 copies of the DNA. Those are the older original copies. They're called parent strands. And then we will make a complement strand for each one of those parent strands, which will be the daughter strands. With the first strand, as we unwind it at this area called the replicating fork, well, the strand will lead in and it'll be a continuous copy. But on the other, and that's called the leading strand. But on the second strand, it has to move backwards. So it waits till it opens a little bit, then starts to copy it backwards, then waits and starts to copy it backwards. And this is called the lagging strand. And those little fragments that it makes are Okazaki fragments. You're going to use a number of different enzymes for this process. Helicase will unwind the DNA. DNA polymerase will build the new DNA units. Ligase will link the little Okazaki fragments. We're now going to look at mitosis, the asexual cell division. Mitosis is the division of DNA. We're going to separate the DNA. This is going to be broken up into 4 cycles. This is a continuous process, but we have broken it up into individual cycles. Prophase, metaphase, anaphase, and telophase. At the end of that, we will have divided the DNA completely, and then we will do splitting of the cytoplasm. This is called cytokinesis. So the 2 individual, identical daughter cells have the same amount of DNA, but they also have enough of the organelles for survival and enough cytoplasm for survival. Now look at this figure of interphase. When you look at the little cell to the right side of the diagram, during interphase, your nuclear membrane will be intact. The nucleolus will be present inside the nucleus. DNA is in that chromatin phase, that stretched out, relaxed phase. And the centrioles are going to be located at one end in a little spot near the nucleus, but they haven't moved at all. Then we're going to double our DNA during the S phase. That's towards the end of interphase. And then we will enter into mitosis. Now we're going to look at the figures of each cell as it changes as it moves through the phases of mitosis. Now in prophase, we're moved from the interphase where the nuclear membrane was intact. The nuclear membrane is now breaking down. The nucleolus disappears. The DNA, which was in the chromatin phase during interphase, will now start to condense into chromatids. And they will form sister chromatids when they partner up and are held together by the centromere, the belt. Now, what they're going to partner up is, remember we doubled the DNA. So I had chromosome 1 from your mom, and I had made another copy of chromosome 1 from your mom that you have in your body. So now these 2 are identical. They have all the same genes, and all variations of the genes are the same. And they are going to partner up, and they are identical sisters. And they're going to put a little belt around their waist called the centromere. And this is so those 2 will travel together now as a group, a pair of 2. Okay? So you're going to have now 46 sister chromatid pairs because we had 23 pairs of chromosomes, 23 individuals from the mother, 23 individuals from the father. So now we've copied each one of those, and we're going to have 46 pairs of sister chromatids that are all partnered up during prophase. So trying to remember prophase, think of prophase starts with P, and partnering starts with P. Also during prophase, your centrioles have gone to the opposite poles, and now they've started to make the mitotic spindle. These are going to be strings that stretch out from the poles towards the center of the cell, and they're made up of microtubules. Then we're going to move in, at this point in prophase, before we move, we're not organized. We have it milling around this whole cell with no nucleus, a bunch, 46 partners. They're all over the place. They're randomly placed. There is no organization. In metaphase, we are now going to call all those sister chromatids to line up in the middle of the cell. They line up on the metaphase plate. The metaphase plate is perpendicular to the centrioles, so we are lined up on the metaphase plate, and those microtubules are going to stretch out and touch us, that are making up the mitotic spindle, and they will actually touch a certain spot on the centromere and attach to the sister chromatids. Each sister chromatid will be attached from one side. During anaphase, the sister chromatids are going to be able to start to move apart because the mitotic spindle is going to start pulling them to the opposite poles. So we're now separating the different chromatids. At this point, a sister chromatid is now considered a chromosome. The difference between a chromatid and a chromosome is that the chromatids share those centromeres, and a chromosome has their own centromere. So now they have separated to the opposite, heading towards the opposite poles, and we will enter telophase. Telophase, we have two nuclei will form. These are going to be at the opposite ends of the cells. Now we've taken those 46 pairs, and we sent a copy of each one of those pairs to the opposite poles. Now we round them up and encircle them with the nucleus membrane so that they can't get away. So we now have two separate nuclei filled with genetically identical material. At this point, we have two nuclei in one cell, but during telophase, we are going to start to do cytokinesis. And this is going to be done by the cell pinching in, and as it pinches in, it will form what's called a cleavage furrow, and this will eventually completely close and separate the cells into one cell into two cells. This is also going to separate the cytoplasm. So we've split the cytoplasm, and we have a nuclei now in each of the new cells. These are called daughter cells, and they are genetically identical to the parent cell.